Humans, including doctors, are extremely curious, and there has traditionally been quite a big role for serendipity in medical progress, hasn't there? Is this compatible with the idea of being very scientific, going from bench to bed to bite?
Yeah, I indeed think so, because you just have to look up for the serendipity, and the serendipity is sometimes, why didn't it work? And then when my students come to me and say that, oh, this experiment didn't work, and I said, that's interesting, why didn't it work? Is it and then something else can come up, and then it's a serendipity.
And if you understand the science, then you get a handle on it.
Exactly.
You know why.
It's about travelling with eyes open and head up, isn't it? And it's trying to understand the negative as well as the positive. But I think such is the quality and breadth of our science that we're reducing serendipity. Of course, blue sky research is utterly important to us all. But by working together, by doing some very large experiments, then it increases the possibility of us sorting through this incredible amount of genetic and other information that you've mentioned, for us to apply it clinically to really simple, practical tests that will make a difference in the clinic. Michael and I are applying molecular tests now to say, this group of patients we'll treat in such and such a way, this group of patients that will not. So it's the epitome, it's the acme of personalised medicine.
I wonder if you could remind me of some of the big successes in the past of molecular factors that have really scored big on the therapy front, and give us some hot tips for the future.
From my field of breast cancer, of course, the Herceptin is one of the big hits, and that this is sort of the model for how we can do it. Because it's not, we don't also know that there are people that should respond that do not respond, and again, we have to understand. But I think this is moving forward from one to the other one, and this is when you put these things together, again, that it's not obvious to treat exactly where you have the biggest hit. It may be that you have to go up and down in the road to find where is the best to do it.
So what new ones do you think are the hottest tips for Stockholm?
It will be definitely pathways. How do you hit the different pathways to have specific drugs for the specific defects that you find?
Which ones?
Well, there will be many in the which will be presented, and I think that we have one, for example, on DNA repair, and how do you add on? How do the cell repair? Can you sort of help the cell to repair, or can you actually make the cell more vulnerable for these type of damages? And it will be other pathways.
I think our interviewer is looking for stock tips. We need to be very careful here.
Well, yeah.
Yes. Yes. Colorectal cancer is a happy hunting ground for markers, isn't it?
It's fantastic. So clearly we have the whole story around mutational KRAS and selecting patients who may or may not benefit from a drug called cetuximab. I think the hot tips are around. I think there's some really nice work coming forward in RNA signatures at last. The whole field has been bedeviled by small studies that nobody can repeat, and now people are doing some really beautifully well conducted and controlled science. I think microRNA is going to be very interesting because much of that's non-coding. And I think in Michael's field there's some lovely mutational stuff in lung cancer.
Well, actually what we see is also a good possibility to combine imaging results, as Anne-Lise has pointed out, with molecular imaging results, with markers which come directly from the tumor or which directly come from blood tests. And that is an emerging field also, particularly for localized treatments, but also for systemic treatments. And we will see a lot of new correlative studies, which then can go into hypothesis-driven clinical trials.
And I think this is also very important at the end when you start to treat, for example, in a neoadjuvant treatment, and that you can follow. And we know that tumors are heterogeneous, so there are many different clones. Yes. And then you start to hit with one drug, and then you can see this is the clone that disappeared. Now we have to do the next drug, and then maybe the third drug. And this way we have the tools now. We have the molecular tools and we have the imaging tools to follow the patients.
Now, Anne-Lise, I know you're doing things like immunotherapy, angiogenesis, of course, which seems to crop up everywhere in cancer, DNA repair, the microenvironment. Give me just a flavour of some of those issues in your track.
Yeah,
in your plans.
It will be, of course, translational tracks. It will be focused on of course, immunotherapy and DNA vaccination. And of course, in angiogenesis, we will hopefully have some of the results from the trials ongoing, and also maybe at the molecular level as well, why doesn't it work always, and when does it work? And then when you go to the microenvironment, then can we target the microenvironment?
Can you?
That's a question, and we will have a panel debate about that. Is it important or is it not important? So that will be, and one of the speakers in that session I talked to, and she actually called me and said that, well, you know, I'm speaking for, but I'm actually not have evidence that it may be also against. So that is against, maybe it is worked for some patients at some time. So that is again targeted and personalized.
And there really is scope for DNA repair, is there?
And DNA repair, definitely. And how do you use or utilize DNA repair for new type of treatment? And I think that these will be examples, and these are some of the highlights, but there will be many others also during the Stockholm meeting on this.
Translational research actually needs a lot of good communication skills between basic scientists, clinicians. It needs to, what David said, it needs a fresh mind to see, oh, there's something unexpected. We have to look, why is that? And then a clinical team, for example, making an unexpected observation, has to bring that back in an understandable way to basic researchers to really work on that.
Yeah, and if you sit together, because then and you have this problem, then you suddenly see the association that you don't, the clinician doesn't see, and the basic science see, and the basic science doesn't see, and the clinician see, and then you communicate like this.
But it's power, isn't it? So there's power of intellect that you've both brought, but there's statistical power, and it seems to me that one of the things that we can do as clinicians, you've done this very beautifully in your breast cancer work, is for us to use our trials, sometimes of thousands of patients. This is the sort of thing that, these are the size of experiments required to validate some of these markers. We can return to you thousands of patient samples, which have got very well characterized in terms of what we'd call phenotype, in terms of how the tumour or the patient behaves. And that seems to me power of intellect, power of number, and that's where the clinical community can really get behind personalised medicine. We've got to move beyond studies of 10 or 100 into studies of thousands of patients, and that can only happen at ECCO. It can only happen in a group such as we three.